presently in Oxford, but I was in Germany and France. Had the honor to be in Italy as well. There was a Nobel Prize winner there, a woman whose name I can't remember, but she was the reason they started that institute. Australia, where they did some state-of-the-art work using multi-agent systems to design actually military applications for, which I didn't really like to participate, and I didn't participate, but I was there more, theoretically. Well, I was actually there, not just theoretically. And then Sony Corporation, and also in Hamburg. So my question that I'm interested in is much higher level. You might think it's maybe too early, given the very interesting, detailed work that you people do. And I must say I'm very impressed with the work that you do. But my question is, okay, how do we get from the genome cell to the embryo, and then also in evolutionary questions? In terms of, I've given this slide before, you may agree with it or not, but I see systems biology as using mathematics, physics, analytical methods to understand biological systems, whereas synthetic biology, we actually construct them from basic components. Systems biology uses computers to model and simulate dynamics. Bioengineering methods are used to design and model and construct these components. And then systems biology handles complexity by abstraction and hierarchy, whereas I think in synthetic biology, we probably would use modularity and minimization techniques. Of course, there's an interaction between the two fields, and it'll be more and more so. And I think synthetic biology uses two basic methods: bottom-up, where you design and engineer the components, and top-down methods, which Professor Blattner will be talking about. And then, of course, there's the mixture of the two, where you just pick and match, like they're doing at what Hamilton was talking about yesterday. Okay, the idea that the basic idea that I work on is developing in silico MCOs, and MCO is a multicellular organism. And then the idea then is to relate the in silico DNA with the natural DNA, where if the in silico DNA generates some multicellular structure, if we have the appropriate code, then we could actually develop a similar structure in the natural organism and vice versa. Now, if we look at natural in silico and synthetic MCOs, you can view the in silico genome as that's the one designed on the computer, generating an in silico MCO. That is used to either understand or to generate a synthetic genome, which then generates a synthetic MCO. And in order to we can reverse engineer, hopefully, the natural genome into the in silico genome, which is then translated into the synthetic genome to generate the organism. It's very futuristic, but I think actually we're quite close, closer than we might want to be. This, again, it looks at it a little bit more, describing what the relationships are. So we have a translation relationship between the in silico genome and the in vivo genome. So because the in silico genome may be a rather abstract formal language, whereas the actual natural in vivo genome will be basically DNA. So in silico development, we simulate the physics, the cell interactions, the cell communication. The genome networks are all simulated in the computer to generate a space-time event, which is then the generated MCO. Then that should be, if we are correct, if we do understand the in vivo natural genome, there should be a nice correspondence relationship in the resulting phenotype. That would be, I think, the ultimate test, a kind of a Turing test for multicellular organisms. If we can generate, say, my idea was to generate, say, a letter on top of a C. elegans worm. Of course, I would generate the W, whereas Blattner would generate the B. But whatever we generate, that would be a proof of concept that the translation relationship that we have established between the in silico and the in vivo genome is accurate. So here's just an example of One of the, it's a bilateral symmetric MCO that was generated with software. It's not just an animation, it's actually generated using genomic networks. So in systems biology, has been a lot of emphasis on modeling single cells, where you try to minimize the processes so we can understand them. Then you model different cell types, look at the pathways, etc. The synthetic systems biology, there's an interplay then between the in silico minimal cell and the in vivo minimal cell that we're trying to generate. So we can design some of the cell networks in silico, and then hopefully they will work in a similar way. People have been doing this for quite a while now, at least for sub-networks, not for the whole network. But as Ham, if I may call you Ham, was talking about yesterday, that is actually getting closer and closer. There's a problem, though, with just modeling single cells. The single cell's lonely. There's no cell interaction. There's no developing tissue. There's no modeling of multicellular diseases. One of the things that we're interested in is modeling cancer. In other words, looking at the kind of regulatory networks that are responsible for pathological multicellular states, which I view as another form of development. And other people do, of course, too. Now, another level of abstraction in systems biology, and my colleague Dennis Noble, who I work with closely in Oxford, you model entire organs. What there is, you view the cell not necessarily as an individual cell, you view it in points in space-time using differential equations, and you model the interaction of these components. The problem is that there's no genome, there's no development. However, of course, very interesting dynamics and very interesting applications, say for studying heart attacks, drug interactions, and that kind of thing. So in multicellular systems modeling, you actually are going after the whole thing. You're trying to get a view of the entire holistic process of how this works. One of the differences when you're doing multicellular development is that we have these weird phenomena where single base pair changes can result in rather radical effects. And here's a famous experiment. Who was it? Was it Lewis that did this one? You guys would know. Anyway, it changes a two-winged fly into a four. Huh? Yeah, it was Lewis, yeah. Into a four-winged fly. Well, in silico, here's an example of an MCO with cell signaling that developed into this little structure here. A single mutation can actually make it then look quite different. As you can see here, we have the two red areas which are not in the original one. So you can get homeotic mutations. Now here's a more theoretical point. How many people have heard of Kolmogorov? Just a curiosity. Okay, hey, okay, Kolmogorov, founder of probability theory, or at least on the foundations of probability theory. Anyway, Kolmogorov was interested in randomness and how to represent randomness. Imagine we have an organism that has this kind of genome, but it looks like this. That would be very strange, wouldn't it? Somehow, it wouldn't seem to me to be possible to get this kind of complex morphology with such a simple genome. Some people, however, there are, I call them New Age people who somehow think that the interactions of proteins in the cell, even if you have a rather redundant genome, will still magically, you get a complex organism. I don't think it's possible, but of course that's an empirical question, whether the analog information in the cell is sufficient to develop such complex organisms without the information being in the genome. Okay, another interesting question is, how is evolution possible at all? Here we have background noise in the environment. It's a problem, because a lot of people say, well, look, the environment is really the cause of the morphological complexity of the organism. Well, to some extent, yes, because, if you can have different phenomena like heat shock, where that influences steps in development, but really, it doesn't it doesn't help us understand the ultrastructure of the organism. So there are some limits, and I call this the complexity conservation principle, where the complexity of the developing organism is really less than or equal to the complexity of the genome, the IAS, which is the interpretive executive system that interprets the genome, and the environment, which is omega here. So some people disagree with this In fact, von Neumann, in his original work on self-reproducing automata, he said that there is a point where living systems reach a level of complexity where they become self-complex. Although I'm a great admirer of von Neumann, and I think in that matter, in this particular matter, I don't know if he is correct there. Okay, there's another interesting principle, which is these are principles which help us guide abstract principles, which may help us guide us to understand how the genome ultimately functions, looking at it very abstractly. And that is that the organism, in some sense, the complexity of the organism has to be actually reflected in the genome and the IES, the interpretive executive system, and the environment. And I would argue that the dominant information has to be reflected in the genome. So, okay, now we're getting to the original talk, the paleogenome. If this reflection principle is correct, then it may help us understand genomes of very ancient organisms. So basically, we model the early development of these organisms and then use the pragmatic semantics of natural genomes to reverse engineer them or understand them. Another interesting concept that is necessary to really understand genomes, I think, is to distinguish between state information, which is like open the door, true or false. See, in the history of logic, the entire last century was basically determined by logicians who thought about things being true or false, right? I mean, they're worried about their arguments being correct or incorrect. But when we start modeling the social world, to my surprise, that most of language, or much of language, actually has nothing to do with truth and falsity, but has more to do with directives, or requests, like open the door, please, or etc. And just as an aside, I helped develop, or actually developed the semantics and pragmatics of these other kinds of speech acts, where I actually looked at the state of the agent, how it's possible to get a kind of a semantics that helps explain these sorts of utterances. Anyway, but the hypothesis here is that most of the genome contains this strategic control information, and it's not so much about descriptive information. Okay? So here's another thing. So parts genes produce protein parts, and of course these protein parts, as we saw in this symposium, that these parts are very interactive. So I have to slightly modify what I say here, but I do say basically, you know, a house in an office building or a bridge, etc., they may have the same components, say all being made of cement and metal, but their architecture is very different. So the difference is how the parts are put together, and that again is not truth and falsity, that's control information. So here we go, scenes. Control genes regulate cellular actions and cell interactions, and these control genes combine to form strategies that are implemented as control networks. And I call these scenes for control gene networks. And then what's nice thing, once you have this notion of control, then you can link them together to make more and more complex scenes. Now the cenome then is the overall control architecture of the genome. So the hypothesis is that the cenome is the dominant part of the DNA in genomes. The second hypothesis is the cenome makes up most of the non-coding regions. Now we've had a lot of people talk about the non-coding region already, and I like the work by the person from Stanford yesterday. We was talking about the interaction between protein and RNA, because I think there's probably the key to control. So a genome, in order to understand genomes, it has to be interpreted. And this interpretive executive system interprets this control information in the genome and executes that information to generate the intracellular and external cellular actions. So this interaction, that little X is the interaction between the IES and the genome, co-evolved in the context of a dynamic environment. So we have to look at the IES as evolving in conjunction with the genome. It's a co-evolution. In the context of the IES, it's seen as more like Mendel's original notion of a gene generating phenotype. Because, you know, let's face it, proteins don't really generate phenotype. Okay, they do if you knock out a protein. It may generate a phenotype, but it's often a loss of function. Often it has no effect at all. Whereas the network conception, which I think is becoming dominant anyway, seems to explain that. So I think this separation of control information from the IES makes possible the evolution of organisms and the variability. Because the IES, once it's kind of set, once this interpretive mechanism, which can be highly complicated and sophisticated, as we saw yesterday, once that's relatively set, it's going to be hard to change that without really having very deleterious effects on the development of the organism. So, but what is not so set are the control aspects of the genome. So here's just some examples of some MCOs. These are all these two pair by pair are one mutation apart. These are one mutation apart. They're one mutation apart. Here's kind of a summary. So we can go through mutations along the line here and go all the way down to where we get something that looks quite different from what we had at the top. And those are all very small mutations that result in rather radical effects. So, steps toward a, I don't know how much time have I got, whatever. I don't, it's like, I should have about 15 minutes or so. Am I over?
We started at 8:30, you got 30 minutes.
Oh, we didn't start at 8:30, but okay. I'll conclude with this. I won't go into the paleobiology stuff then. So the steps toward a synthetic MCO, first we have to do this decoding problem, which I think will be central. And I think the ENCODE project will help with that. This will lead to an understanding of these scenes in the genome. And once the synthetic genomes, then once we can do that, and once we have what Hamilton is working on, if we can then insert this sort of designed genome into the cell, then the thing that's designed in software should be able to be replicated then as a multicellular structure. So then the line between life, in silico life, and synthetic life will vanish. And that's basically the point. And then I go into the rest of the talk is because it was really for paleontologists looking at how we can decode the original thing that happened in the Cambrian and Precambrian, etc. Here's an interesting one. Here we've got the same genome, but it's because the IES is different, we've got a very different phenotype. Very, very strange, but interesting effect. So in the Precambrian, I'll just conclude that the Precambrian was much more flexible. There was this coevolution going on. Genes basically don't evolve that much, and if they do, we usually have disastrous effects. So let's go to the end, and that's it. Thank you.